Welcome to the fourth video in the Bacterial Genetics unit. In this video, we're going to talk about regulation of gene expression. So last video, we looked at how you make a functional product from a gene, so mainly the gene expression through transcription and translation. In this video, we're going to talk about how you control that expression. So when do we make certain proteins and how much of those proteins are made? That control is based on the environment that the organisms are found in. There's also additional videos on the lac operon and this regulation on Canvas for you, because the regulation of lac operon tends to be one of the more challenging subjects for students to understand. Okay, let's go ahead and take a look. The functions that a cell possesses are controlled by the proteins that are present in the cell at any particular time. But it's important to recognize that not all of the genes in the genome are transcribed and translated at the same time. Both transcription and translation require ATP, so they require the cell to use energy. This means the cell doesn't want to waste energy making proteins that don't have a function right then, that are just going to sit in the cytoplasm and do nothing. For example, if a bacterial cell has a set of proteins that allow it to grow better at warmer temperatures, it's only going to want to produce those proteins when the temperature gets warmer. It doesn't want to bother making the proteins at cold temperatures because they won't have a function for the cell. This control over gene expression is called gene regulation. In eukaryotic cells, gene regulation is controlled by the environment and the tissue type that the cell is part of. This is why your liver cell and your heart cell function very differently, even though they have the same genome. It's because different genes in that genome are being expressed in those different cell types. For bacteria, they're single-cell organisms, so their regulation is really based on what environment are they living in at any one time. We can actually see an example of that when we look at these pictures over here of Serratia marcescens. This is a bacteria that produces a red pigment, but it only expresses that gene when grown at cooler temperatures, at room temperature. If it's grown at the body temperature at 37 degrees Celsius, the expression of that gene stops. So this is regulated by temperature, and we can see that on the plates. We can see that at room temperature, those organisms are red in color because the gene is being transcribed and translated to make that red pigment. But on the plate on the right, it's grown at 37 and those are white. That gene is no longer being expressed, it's now off, so we don't make the pigment and we don't see that red phenotype. When you look at methods to regulate gene expression, it can be controlled anywhere along the central dogma. So remember, we go from DNA to messenger RNA to protein. And so we can control at the transcriptional level, going from DNA to RNA. We can also control at the translational level, going from RNA to protein. And then when we have a protein in the cell, sometimes those proteins need to be modified in order to be activated. That's called post-translational control. As we look at the different levels, most cells use transcriptional control more than any of the other types. And this is because this is the most efficient. Because transcription and translation take energy, if we just stop either one of them from happening, so we stop it at the very start of the process, that's going to save the most amount of ATP. However, it's also the slowest, because if you need that protein, you have to take the time to do transcription and do translation before the functional protein shows up in the cell. The opposite is post-translational control. That's the least efficient because you've used all the ATP to make the protein and now it's just sitting there. But it's also the fastest because you could have a non-functional protein in the cell that just needs a slight modification to change it to an active protein, which is much faster to regulate at the post-translational level. Cells do a combination of all of them, but like I said, post-translational tends to be used the least and transcriptional control tends to be used the most. Because transcriptional control is the most common, we're going to focus on that level for most of this video. There are two ways to classify transcriptional control, one based on how gene expression is changing, and the other based on the type of regulatory protein doing the control. We'll first look at the classification based on how gene expression is changing. Induction of genes means that the genes were originally off, and then they get turned on because of an environmental change. What that looks like is we have our DNA, and here's our promoter region, and then we have our genes. And this gene is originally off. And when we mean off, we typically draw it looking like this, and what that means is that there's no transcription happening. So we're not making any messenger RNA, which is why the gene is off. In induction, some sort of environmental change is going to happen, and that causes the DNA from going to being off to now being transcribed. So now we're going to be making messenger RNA from that gene. So we've gone from off to on. Repression is the opposite. So in repression, we've started with a gene. Here's our promoter and the gene. And we've started with a gene that is on. So we are making messenger RNA. The gene is being transcribed. Some environmental change happens, and in response to that environmental change, we've now shut the gene off, so we're no longer getting any transcription from the gene itself. The regulation of expression is mediated by the environment through the effects of regulatory proteins. There are both activator and repressor proteins, which are typically controlled by the binding of small molecules called ligands. We're going to look at those regulatory proteins next. We can describe gene regulation based on induction or repression. We can also describe it based on the type of regulatory protein being used. In this case, repressor proteins are used in negative control for regulation. A repressor protein functions to block transcription and thus repressing the gene and ensuring no messenger RNA is made. They function by binding to regions of the DNA called operators. Repressor proteins can be used for both induction of genes and repression of genes, depending on when they are bound to the DNA. In gene induction, remember that the gene is originally off and then it switches on due to an environmental change. Repressor proteins function to prevent transcription. So this means that the repressor protein was originally bound to its operator. So we can again draw the DNA. We see here the DNA. Here we have the promoter. The operator site is actually found downstream in between the promoter and the gene itself. And remember, the operator site is where the repressor protein binds. And so in induction by negative control, what we're going to look at is here is your repressor protein bound to the DNA at the operator site. And because that repressor protein is there, this gene is off. It is blocking transcription. But then when the environment changes, remember in induction we go from a gene that is off to one that is now going to be transcribed. So now we are going to be able to transcribe. We're going to get our messenger RNA, and you notice that the repressor protein is now no longer bound to the DNA. So the repressor protein was bound, and then it comes off, which allows our gene to go from off to now being on. Typically, the control of the binding to the DNA or not is due to the binding of a ligand to the protein itself, which we'll see in a few slides when we walk through a specific example of regulation with the lac operon. Repressor proteins can also be involved in repression of a gene by binding. So this would be negative control and repression. In this instance, the repressor protein is not bound to start with, so we have our promoter, our operator with nothing bound, we have our gene, and in this case the gene is on, so transcription can happen, and we are seeing messenger RNA produced. But now there's an environmental change, and the repressor protein, which originally had not been bound to DNA, is now going to come over, bind to that operator region, which then blocks transcription from happening. We get no transcription, and the gene is now off. So we've switched from being on to off because the repressor protein went from not bound to bound to the operator. Activator proteins are the opposite of repressor proteins. When regulation is being done by an activator protein, it's called positive control. These proteins bind to activator binding sites in the DNA, and they help recruit RNA polymerase to the promoter. By increasing the amount of RNA polymerase binding to the promoter, activator proteins increase transcription of a gene, turning it on. Similar to repressor proteins, they can be involved with both gene induction or repression. In gene induction, the activator protein starts off not bound to the DNA. And so we have our DNA here. Remember, we have our promoter region, and here we have our activator binding site. Down here is the gene itself. So in this instance, there is no activator protein bound, so the activator protein is over here somewhere floating in the cytoplasm. And this means we're not getting any expression here. So this gene is off, or sometimes it is low expression, where we get a little bit, but not a whole lot. Then the environment changes, and then something happens that allows the activator protein to now come over and bind to that activator binding site. This is going to allow RNA polymerase to come in and bind to the promoter easier, and with more RNA polymerase binding, we get more transcription, which makes lots of messenger RNA, and now the gene is on. So again, this is induction. We've gone from off to on, but it's induction by an activator protein because the protein was not bound. Here, the environment changes and now it can bind, turning the gene on. We see the opposite for repression by an activator protein. So this would be positive control leading to repression. So again, we have our DNA, we have our activator binding site, and our promoter, but in this instance, the activator protein is already bound to that activator binding site, recruiting RNA polymerase, leading to lots of transcription. So this gene is on, but then something in the environment changes. That change causes the protein to come off of the activator binding site, so it's no longer bound. Because it's no longer bound, RNA polymerase is not coming in and binding to the promoter, and without RNA polymerase, we're not getting transcription, and so the gene has been shut off. So again, we've gone from on to off, which is repression of the gene, and it's controlled by the binding or the unbinding of the activator protein. We're now going to use the lac operon as an example of both positive and negative regulation. The lac operon is a set of genes that are involved in the breakdown of lactose disaccharide for energy. Operons tend to group genes together that are involved in the same pathway and thus needed for the cell at the same time. This way, regulation only has to happen at one promoter. The two main genes of the lac operon are LAC-Z, which encodes a beta-galactosidase enzyme. This enzyme cleaves lactose into two monosaccharides, glucose and galactose. So here's the beta-galactosidase enzyme, encoded by LAC-Z. The second gene, LAC-Y, encodes lactose permease, which is a transport enzyme that allows lactose to get into the cell easier. People are not real sure what LAC-A does, so we're not going to talk about it today. Since these genes encode proteins that are needed for lactose digestion, they're only needed if there is lactose in the cell. Additionally, the cell would prefer to use glucose for energy, since glucose feeds right into glycolysis without any additional steps. Thus, these genes are only highly expressed in conditions that contain lactose, but no glucose in the environment. The first level of regulation uses a repressor protein to monitor the presence or absence of lactose. Remember, we only want expression when lactose is present, so when it's absent, we want the genes to be off. This is the role of the repressor protein, which is called LAC-I. So let's see how this regulation is going to work. Here's our DNA. We have our promoter, our operator, which is where that LAC-I repressor protein is going to bind, and then LAC-Z, LAC-Y, and remember LAC-A at the end. So this is our operon. So now in this environment, we're going to have no lactose present. When there's no lactose, we don't want to bother wasting the energy to make these genes into proteins because they wouldn't have any function. So in this scenario, LAC-I, which is the repressor protein, is bound to the operator, which means RNA polymerase can't get to the promoter to do transcription, so we have no transcription and these genes are off. But if the environment changes and now lactose shows up, we're going to want to start being able to express these genes. So again, here's our DNA with the promoter and the operator and the genes of the operon. And now lactose is present in the environment. Lactose gets converted at small levels to something called allolactose, which is just a slightly different structure. So these little red dots will represent the allolactose that's now present. Allolactose is important because when it gets into the cell, it's able to bind to the lac-I protein. And when that happens, it causes lac-I to change shape. So lac-I bound with allolactose is a different conformation, and this conformation is going to pull it off the DNA so it's no longer bound to the operator site. Now that we don't have a repressor protein bound, RNA polymerase is able to come in and it's able to find this promoter every once in a while. And in this instance, we have a low level of transcription. So I want you to notice the gene was originally off. We've now turned it on, but only to a low level. And this is mediated by the absence or the presence of lactose. So lactose shows up, we remove the regulatory protein lac-I from the operator region, and that allows transcription to begin, so we're now getting a low level of expression. The repressor protein lac-I was controlling whether or not transcription could happen from the lac operon based on the presence or absence of lactose. But remember, we only want expression when lactose is present, but also when glucose is absent. And so there's a second level of regulation for the lac operon, which involves positive control with an activator protein called CRP. And so again, we're going to look at what happens with the DNA itself. So we're going to have an environment to start with that lactose is present, so we'll leave off where we left at the last image. And we're going to start with glucose also being present. So we have our promoter, our operator, and now we're going to have an activator binding site. And then we've got the genes of the operon itself. Now, because lactose is present, remember lac-I is bound to allolactose, and it's floating off in the cytoplasm somewhere. So there's no repression happening here, which means RNA polymerase can come in every once in a while and do some transcription. But there's glucose present, which means we don't really want to use these genes yet, so we want to make sure that this is low level of transcription, and that's done because of the activator protein. So besides the repressor protein, there's also an activator protein called CRP. And when glucose is present, CRP is not able to bind to the activator binding site. So without an activator protein, we just have low levels of transcription. But as, say, E. coli is growing, it's using up all of the glucose for energy. Eventually, the environment will change. We still have lactose, but we've used up all of the glucose. And so now we're going to see how that affects our gene expression. Remember, we've got our activator binding site, we've got our promoter, we have our operator, and then we have the genes. So now that glucose is absent, something's going to change in the cell. And that change is going to be an increase in a molecule called cyclic AMP. So when glucose levels are high, cyclic AMP is low, and when glucose goes away, the amount of cyclic AMP increases. Cyclic AMP is what we call a secondary messenger. Mainly, it allows us to regulate the levels of gene expression based on something happening in the environment without looking at that something directly. So glucose is not actually going to be binding to the activator protein or not. It's going to be cyclic AMP that does the binding. Remember, we had our CRP protein, and now that cyclic AMP has increased in the cell, cyclic AMPs are little purple dots, cyclic AMP is able to bind to CRP. That changes its conformation, and allows it to bind to the DNA. So now CRP with that cyclic AMP bound is able to bind to the activator binding site. It functions like an activator protein, recruits RNA polymerase to really high levels, which means now we get lots of transcription, and now we're seeing high levels of transcription or expression of the genes. So when glucose was present, we had low levels because we did not have the activator protein binding, but when glucose goes away, cyclic AMP levels increase. Cyclic AMP binds to the activator protein, which allows it to bind to the DNA. This recruits RNA polymerase, leading to high levels of expression, which is important because we now need these proteins to break down the lactose and get energy because there's no more glucose to use for energy production. In the lac operon, we saw that small ligands such as allolactose or cyclic AMP can bind to regulatory proteins, changing their conformation or their shape, which controls if they could bind to the DNA or not. Those small molecules were one way that the cell was able to sense and respond to changes in the environment. Two-component systems are another way that cells are able to affect gene expression based on environmental changes. In a two-component system, the environmental change or stimulus never actually enters the cell. Instead, it binds to a receptor protein, which is usually in the membrane and is typically a kinase receptor, that's the first component of a two-component system. The binding of this molecule to the cell indicates something in the environment that then causes a change inside of the cell itself. That change is typically the addition of phosphate groups to a response protein, and the response protein is the second part of that two-component system, and the addition of the phosphate group causes the response protein to change conformation, which allows it now to bind DNA. This is now a functional regulatory protein, and the conformational change allowing it to bind DNA is similar to the conformational changes we saw when we talked about the lac operon.